I'd like to start by thanking as well Brett and Philippe for inviting me to this symposium. And I consider myself more of a developmental immunologist, but I think that getting to be part of this great symposium has really opened my eyes to some of the aspects of mucosal immunity that I think is going to be very useful for trying to integrate some of these developmental questions into the question of how these innate lymphoid cells are actually involved in some aspects of homeostasis and disease protection at mucosal sites and maybe at other sites. So the outline is here. I'd just like to review with you some of the aspects of the discovery of these different innate lymphoid cells and try to put them together in a context where we think they actually are related by virtue of expression of some transcription factors and cytokine dependencies, and therefore define a family of cells, lymphoid in nature, that have an early response in immune responses, the early reactivity. Following that, I'd like to compare these ILCs with more classical T helper cell subsets, try to give some ideas about the way we think about studying them, about their roles, how they might have evolved, and get some differences. And then in the last part, some unpublished data on some approaches we're trying to take to dissect ILC biology using conditional gene targeting, specifically in cells expressing these NK markers, so classical NK cells and these IL-22 producing NK-like cells, which also leads into questions related to the biology of these cells and their stability and plasticity in terms of phenotypes and functions. So we're interested since some time in NK cells as a lymphocyte subset that has innate properties. And of course, NK cells are positioned here as links between the innate and adaptive responses. The NK cells, of course, were discovered based on their natural cytotoxicity. So one could ask how that is actually important for some relationship to adaptive immunity. Of course, in terms of immunosurveillance and immune defense, NK cells could play a direct role. But now it's also appreciated that the fact that NK cells can lyse infected and stressed target cells, thereby providing antigenic substrates for dendritic cells, clearly implicates NK cells as well in priming of adaptive immunity. NK cells are also very strong cytokine producers. They can modify target cell expression of class 1, class 2, and in the end, thereby can interact and influence adaptive responses. So clearly they're placed in between this innate adaptive axis, and we've been trying to understand the developmental biology and how these different functions in these innate lymphocytes are acquired. So I won't go over the whole scheme of development of NK cells. This has been work from many groups, including our own. But the point here is just to say that these cells derive from hematopoietic precursors and go through a series of developmental intermediates, which include precursors that are committed to the NK lineage, which then receive signals that allow their maturation, which includes, of course, acquisition of many different cell surface receptors. I don't think it's really important to go through all of these. This is driven by cytokines, so IL-15 plays a very important role in the differentiation and acquisition of effector functions, but also in the maintenance of these cells as they're developing. Now, another aspect of innate lymphocytes is the fact that the effector functions that they exert, so for the NK cell, there's cytotoxicity and cytokine production, they're acquired very early in this process. So using reporter mice, you can actually start to demonstrate expression of perforin, for example, or interferon gamma, very early in the process, even at the point of the earliest committed progenitors. So this is somewhat different from T cells, which of course acquire these through differentiation. These innate lymphocytes have these pre-programmed in their development, and they're ready to go as soon as the cell is coming out into the peripheral lymphoid tissues. Now, one important transcription factor which is involved in the generation of the NK lineage is a protein called NFIL3 or E4BP4. This transcription factor has been shown to be critical for the development of the NK lineage. In the absence of this factor, there's complete ablation of NK cells, whereas basically all the other hematopoietic lineages develop completely normally. Now, NF-IL3 is linked to ID2, which is a transcriptional repressor, and NF-IL3 is thought to induce ID2. ID2 is also critical for NK cell development. In the absence of ID2, you have a complete absence, again, of the complete lineage of NK cells. Now, how does that work in terms of lymphoid development? This is actually very interesting and very nice because we know that deriving T and B cells from hematopoietic precursors involves an increasing activity of EBOX transcription factor activity. This is involving E2A and E47 and HEB in the T cell lineage. And this ID2 is actually a repressor that can then inactivate these E protein activity and therefore divert then the precursors into the NK lineage. So ID2, its expression, when it's controlled in a fashion, can actually switch the lineage between T and B cells, so the adaptive arm, into the NK lineage or the innate arm. So this is a molecular switch which was important for distinguishing two subsets of these different types of lymphoid cells, innate versus adaptive. Now, ID2 deficiency also has other effects, and one of the effects in the hematopoietic system that's manifest in the absence of ID2 is the lack of structured lymphoid tissues. And this is the original publication from Yokota and Gruss, where they were able to show that the absence of ID2 clearly affects development of peripheral lymph nodes here in the abdominal wall, in the mesentery, and here in structures in the intestine, including the development of Peyer's patches. And we know now that this is due to interactions between lymphoid tissue inducer cells, which express ID2 and are able then to interact with stromal organizer cells to provide the initial interactions necessary for recruiting in lymphocytes into these structures. So we know that ID2 plays a critical role in the development of these LTI cells, and these are cells, as has been shown by Nishikawa initially, are expressing IL-7 receptor. Mebius and colleagues showed that they are CD4 positive and CD3 negative. So these are another subset of innate lymphoid cells that depend on ID2 in order to generate a critical effector for the development of the immune system. Now, a link between the role for LTIs, at least during the fetal period, this was nicely summarized by Gerard. We know that they're important during fetal period for the generation of these different lymphoid structures, but it's been more recently appreciated that LTI cells also play a role potentially in the adult as a source of cytokines that are important for immune defense. And this is the report from John O'Shea's group, where he was able to identify these LTI-like cells in the adult, and here in the spleen, again, CD4 positive, CD3 negative cells. And under appropriate conditions, stimulation through IL-23 are able to produce quantities of IL-17A. These are also cells that, like LTI, express RR gamma. They express, of course, the IL-23 receptor. And through this interaction of IL-23 with LTIs in the adult, we have another means to produce important cytokines involved in inflammatory processes and also in tissue repair. So the relationship between LTIs and NK cells is, of course, suggested because of the importance of ID2 in the development of both of these innate lymphoid lineages. Furthermore, Kay's group has shown that a transcriptional activator that was normally identified in thymocyte development called TOX also is critical for both the development of NK cells and LTI cells. So there's a clear link between these two innate lymphoid cells in terms of transcriptional profiles. But nonetheless, these cells diverge in terms of their function, clearly, and they also diverge in terms of other transcription factors which are important for their development. I mentioned the NF-IL3, E4BP4 for the NK lineage, and RR gamma, which was shown by Gerard when he was with Dan Littman, is important for development of the LTIs. In addition, the cytokine profiles of these cells are quite different. We know that NK cells are highly IL-15 dependent. LTI cells do not depend on this factor, whereas LTI cells, in contrast, express the IL-7 receptor and require IL-7 in terms of their development and homeostasis. So while these cells, these innate lineages are related in terms of transcription expression and specifically linked by expression of ID2, they're clearly divergent in terms of their effector functions, in terms of cytokines that are important for those effector functions and for their maintenance in the periphery. So we've seen this slide in Fiona's talk. It's a version of that. This is from a review that Harrigan and I wrote in Nature Immunology earlier in the year. And we're proposing that this ID2 expression in precursors actually then defines what we call the innate lymphoid cell family. The founding member of that family is the NK cell, which under the influence of cytokines produces cytotoxic and gamma interferon producing innate lymphoid cells. These we call ILC1s by analogy with T helper 1 subset of T cells. In addition, we have this RR gamma positive population. Here they're going to be coded in yellow, which under the influence of IL-7 gives rise to LTIs, which have important roles in fetal development in terms of lymphoid tissue generation, but also in adult by virtue of some cytokine production. Now, we kind of fell into this question about RR gamma positive innate lymphoid cells when we were looking to survey NK cells in other tissues. And in order to do this survey, we made use of what we thought was the best NK cell marker at the time, and this is a natural cytotoxicity receptor called NKp46. This is a family of cytotoxicity receptors expressed in human cells and identified by the Morettas initially in the human system. One of them is conserved in the mouse. It's the NKp46, and it signals for activation of NK cells through ITAM-coupled receptors. It's highly NK specific. It's not expressed in NK T cells or in conventional alpha-beta T cells, and as such it's been proposed as a marker for NK cells. So Ofer Mandelbaum in Jerusalem had made a knock-in mouse where he replaced GFP into the coding sequences, and then using this reporter mouse, he was able to show that NK cells, which are GFP here, are able then to be visualized, and we've collaborated with a number of people, including Philippe Sansonetti at Pasteur, to look at the behavior and the biology of these cells in situ. Now, we started surveying different tissues, and one of the tissues that was on our list, of course, was to look at mucosal surfaces, and that's when Naoko Sato Takayama came to the laboratory. She had been trained in Tokyo in Hiroshi Kyono's lab, a mucosal immunologist, and she decided to start to look for these cells in the gut. And when she used these NKp46 GFP mice, she was able to verify that, yes, you get expression in NK cells in the spleen and in many other tissues like the bone marrow and thymus. I won't show you that data. But when she looked in the gut, it was clear that these GFP-positive cells had different phenotypes compared to the classical NK cells in the spleen. While you could find some NK cells that had classical features expressing NK11 and many other markers that we know that are expressed by NK cells, there was a large population of IL-7 receptor positive cells that lacked NK1, but that were also expressing NKp46 and resident in the small and large intestine, enriched in the lamina propria, but also found to some degree in the intraepithelial compartment. So I won't show you all the data, but just to summarize these findings, Naoko went on to show that, in fact, these atypical NK cells had actually very few NK cell markers. They didn't express any of the receptors that we normally associate with NK cells, which are important for either activating or regulating the function of the NK cells. Moreover, they didn't express any of the effector functions of NK cells, including perforin or gamma interferon. But in contrast, they were highly expressing RR gamma T, and when stimulated by IL-23, were very strong producers of IL-22, specifically IL-22, no production of IL-17A. So they seem to be a subset of cells that was very much tuned and poised for IL-22 production in the gut. And as such, we've renamed these NCR22 cells. They fall into what we can consider now as a subset of ILCs that express IL-22, or an ILC22 subset of cells. And again, they have a phenotype which is quite distinct from the classical NK cell in terms of transcription factor expression, cell surface marker expression, and production of cytokines where here we have IL-22, whereas the NK cell, of course, is tuned towards gamma interferon production. So we have a series of ILCs now that are found in mucosal tissues, including LTI cells, which can produce IL-17, IL-22, these NCR22 cells, which express an NK marker and appear tuned towards IL-22 production, and the NK cell, which is a gamma interferon producer. So the question then becomes, are these cells related, and how are they actually derived? Do they have some plasticity in terms of their biology, or are these distinct subsets of different lymphoid lineage cells? Well, the NKp46 positive IL-22 producers as well express ID2. So again, these are part of this family, which is now emerging, that depend on ID2 for their development from hematopoietic precursors. These cells express RR gamma and depend on IL-7, so in some ways they are more closely related to LTI cells than to the NK cells to which they share this one particular natural cytotoxicity receptor. Moreover, IL-15, which is critical for NK cell development, does not influence the absence of IL-15 or its overexpression does not influence the development of these NCR22 cells. So clearly, these unusual IL-22 producing NK cells appear more related to the LTI lineage. But are they really, in some ways, functionally associated with LTI function? That appears not to be the case. At least initially, it was thought that these cells sit in cryptopatches. Some of that was probably artifact due to the antibody use that was in those particular experiments. They used polyclonal antibodies, which actually had very poor specificity. We revisited this notion using mice where we had knock-in alleles for either NCR1, so this GFP expressed in the NCR1 locus, or RR gamma expressed in the GFP expressed in the RR gamma locus. And in both cases here, you can see that you have an abundance of these RR gamma positive cells here in red, here in green, but quite an absence of the NKp46 positive cells, so very few NKp46 positive cells expressed in these patches, which are pretty much by definition the cryptopatches that are the precursors for those isolated lymphoid follicles. So while initially it was thought that these cells might be related to LTI and sitting in the same types of structures, in fact, these IL-22 producing cells are not in the cryptopatches and are quite distinct from where the LTI cells are. These cells sit mostly in the lamina propria, where we believe that their production of IL-22 is relevant for a crosstalk with the epithelial cells that are expressing the IL-22 receptor. Now, Gerard has made a very nice study looking at the relationship of these different RR gamma positive innate lymphoid cells, and I just want to make a few points for this particular study because I think it impacts on the idea of the relationship between LTI cells and these IL-22 producing cells. And Shin was able to look at the culture of the individual RR gamma positive ILC subsets in vitro. And one of the findings from that work was that when you isolate these individual ILC subsets and you culture them, they remain stable in terms of their phenotype, and they don't interconvert. So when you isolate LTI cells, CD4 positive LTIs, they don't end up acquiring NK lineage markers, so they don't convert to the IL-22 producing NKp46 positive cells. Moreover, when you deplete LTI cells in vivo using anti-CD4 antibodies, you block the development of that subset, but you don't influence the other RC positive ILC subsets. So the NKp46 positive cells that express IL-22 are not affected by this depletion of LTI cells. And finally, you can identify precursors in the fetal hematopoietic tissues, like the fetal liver, that differ in expression of alpha-4 beta-7 integrins, which are expressed on hematopoietic precursors, and different in the level of RR gamma C expression here by the GFP marker, where you can dissociate the development of the LTI-like cell, either expressing CD4 or not, with these NKp46 positive cells, suggesting that, in fact, although they're derived from ID2 positive precursors, these cells develop in an independent fashion and are not interconvertible. So we add another subset here to this emerging family, these ILC22s, which have been described in the human by Marco Colonna and by Harrigan Spitz, and by us and Eric Vivier and Andreas Diffenbach's group in the mouse. These cells appear to be very restricted in terms of their cytokine profile, very much tuned towards IL-22 production. But this, of course, then extends the notion of the innate lymphoid cell family, and specifically the subsets that express RORC that can produce these important cytokines at mucosal surfaces. Now, Fiona just mentioned, of course, in her talk, the discovery from Sofia on the innate producers of IL-17 and the importance of the production of these cytokines in this colitis model induced by Helicobacter. So this subset of IL-17 gamma producing cells expresses Thi1. I just wanted to add the point that, in fact, all of the ILCs that have been described express Thi1. So while this marker is very useful in terms of identifying innate cells that can produce these cytokines at very high levels, the Thi11 expression is by no means specific to these IL-17 producing cells. It can also be, of course, expressed by NK cells, by the IL-22 producing cells, and as well even by some of the Th2 producing, cytokine producing innate lymphoid cells. But nonetheless, of course, these treatment with the anti-Thi11 had a dramatic effect in this colitis model, suggesting that the sources of cytokines from these innate cells are very critical in terms of regulating the pathology that's involved in this particular model. Now, ILC17 cells exist in the steady state as well. They're not only found during colitis, they're maybe smaller in number, but nonetheless, they do develop in the absence of pathology. So clearly we have many different subsets of ILCs that are RR gamma dependent, producing a variety of different cytokines. And of course now the challenge is to try to understand how these cells have specific or redundant functions in terms of immune biology at mucosal surfaces. So just to complete the scheme, these Th2-like innate lymphoid cells were described by 3 groups this past year. They were mostly identified using worm infection models where the transfer of these cells was able to then control the worm infection, allow for expulsion in vivo. So these are very potent cells, and these produce a range of cytokines, including IL-5, IL-6, IL-13. They can also crosstalk with B cells by virtue of the IL-6 production. That's thought to be important for some growth of some innate B lymphocytes. But the IL-5, of course, can play an important role in recruiting other effectors like eosinophils. So this might be as well important in the elimination of certain types of parasites. And clearly the IL-13 production by these cells is very important for induction of mucus at mucosal surfaces and for worm expulsion. So we have clearly a very large panel, and now we see that in contrast to what we thought a couple of years ago, where maybe innate cells were much stereotyped in their response, we see that there's quite a diversity in terms of the cytokine production that's available during immune responses, even at the innate level. So that leads to, of course, some discussions and comparisons between these innate cells and the adaptive pattern of differentiation of T cells from naive precursors. And while it's clear that there are some very striking similarities between these two systems, the virtue of differentiated subsets that can produce type 1 cytokines, type 2 cytokines, Th17 type cytokines, and IL-22, more IL-22 restricted subsets, there are some clear differences between these two systems. The first I mentioned already is that this innate production of cytokines seems to be very rapidly acquired during the differentiation process. These cells are almost programmed to make these cytokines in contrast to the T cell side, where this requires extensive differentiation and environmental factors, which then can tune the different T helper subsets. Another point that's critical here is that while the transcription factors that have been isolated and identified as signatures for the T helper subset are also find parallels in the innate system. For example, GATA3 in the ILC2s, it's not here on the slide, but in fact these cells do express GATA3, TBET and RO gamma for TH1 and the TH17, etc. The difference is that the cytokines that are involved with promoting the differentiation of these T helper subsets, including soluble factors like IL-6, IL-23, IL-12, etc. for TH1 cells, are not required for the differentiation of these innate cells. So when you look in mice that have been invalidated for many of these factors that are critical for the development of the T helper subsets, you still find that the innate cells that can produce cytokines are present and they're still functional. So the wiring of this innate lymphoid cell is clearly different from the differentiation induced and environmentally conditioned response of the T helper subset differentiation. And finally, of course, there are a few T helper subsets here which we don't find yet parallels in the innate system, notably the Treg cells expressing FOXP3. So far, no one has identified an innate cell that expresses FOXP3. It may be out there, it may be induced under some particular conditions, but so far it hasn't been found. And maybe the regulation of this innate system is not going to depend on an analogous type of Treg cell, in that the cells here are relatively short-lived, they're cytokine dependent, so there are many aspects in which they could be controlled indirectly without need for a specific innate lymphoid cell suppressor cell. So just to finish up, I'll talk a little bit about some of our attempts to try to dissect roles for these different ILC subsets, and in order to do that, we've been developing some models using conditional gene targeting. And one of the questions we've been asking is what the importance for this innate IL-22 production is in mucosal immunity. And I think that we already know the answer to this question, and that is that the interactions between the ILCs that produce IL-22 and the epithelial surfaces that express the receptor provide barrier function, and that at multiple levels, including the structure and probably tonic growth of the epithelium, as well as the production of antimicrobial peptides. And the role for IL-22 as a critical factor is been demonstrated in this particular paper by the Genentech group, where they used the infection model of Citrobacter rodentium, and were able to show that the early production of IL-22 is, of course, critical for defense against this particular pathogen. So we've been using this model and trying to look at what cells are producing IL-22, and while there may be some modulation of IL-22 production depending on colonization, and we heard from Gerard about how this might be regulated through IL-25, it's clear that the IL-22 that's being produced in the context of this particular infection is largely restricted to these innate lymphoid cells. Here you can see the IL-22 production after infection is coming from CD3 negative cells. This is highly inducible by IL-23. And if you look inside this IL-22 positive subset of cells following infection with C. rodentium, in fact, you have quite a diversity of cells that are involved. LTI cells are producing IL-22, NCR22 cells are producing IL-22, many cells that lack either the natural cytotoxicity receptor or CD4 are producing. So there's quite a diversity. And we've been trying to find genetic models that can start to dissect the functions of these different IL-22 producing cells. And we've been taking some of the knowledge that we've gained through the analysis of the cytokine sensitivities of these different subsets to start to address this question. So here is a Citrobacter rodentium infection in a rag mouse, and as we know, these mice can resist the infection for quite some time because of the production of IL-22. In the absence of NK cells, so this is a mouse which lacks signaling through IL-15 and therefore doesn't develop any NK cells, there is an effect, but clearly it's much later than what was seen in the initial experiments on IL-22 neutralization. So NK cells are important in the defense. It's probably by virtue of the fact that they produce some gamma interferon, and this is a rather late stage of defense mechanism, but nevertheless the NK cells are involved. Now initially we used rag gamma mice to see what the effect of the sum total of these different ILC subsets would be, knowing that in the absence of gamma C you lose IL-15 and you also lose IL-7, so you lose all of these different ILC subsets, NK cells, IL-22 producing NCR positive cells, CD4 LTIs, the IL-17 producers that Fiona's described, and as well the nuocytes. So basically, all of these different ILC subsets are lost in the absence of gamma C. And there's a dramatic effect in terms of the sensitivity of these mice to Citrobacter rodentium. The IL-22 production in this context is extremely low. There's a little bit. If you would compare it to an IL-22 knockout, you would gain maybe a day. So there is some gamma C independent IL-22. But in fact, the majority of the IL-22 is coming from these ILCs, and now the challenge, of course, is to try to understand which of the different subsets is involved. So we've generated these NCR-CRE GFP mice. These were using the promoter for NCR1 linked to a CRE GFP fusion protein, and these mice basically express GFP in the NK lineage. Two types of mice we have made. One expresses in the majority of the NK cells. Another line, actually, we call an NK balancer because there's only about half the NK cells express the GFP. So this is a way to actually look at comparing NK cells that have and have not deleted a particular target in the same animal. We think they're going to be very useful mice for that kind of an analysis. These CRE mice actually can be tested by using reporter mice. Here the rosa-stopped RFP mice, when crossed to a CRE transgenic, will drop out this stop cassette and indelibly mark the cell with red fluorescent protein. You can see here that if you look in the NK cells, the majority of those cells are RFP positive, and if you look in the RFP positive cells in the spleen, they're all NK cells. So clearly this CRE is working as we hoped. It's not expressed in any of the other lineages as far as we can see in the hematopoietic system. It's not in B cells, T cells, myeloid cells, dendritic cells. And this is also using this stop RFP model where it's very sensitive for looking at small numbers of cells which might have indiscriminate kind of expression of the transgene. We don't see that really in any of those lineages. Now, this is also expressed in the gut. If we look at these NKp46 positive cells that we know exist in the gut, again, we mark most of these cells by virtue of this tagging scheme with RFP. And again, both these CD127 positive IL-22 producers as well as the conventional NK cells are marked in the gut by this particular CRE mouse. So we're trying now to find ways to dissociate these two, and of course, by virtue of the fact that they depend on different transcription factors, we think we have the best way to kind of take it apart. So we've been able to get RR gamma flox mice, NF-IL3 flox mice, and to take out both sets, we're using the IL-2 receptor gamma flox mice. These will give phenotypes in which we can have deletion of the IL-22 producing cells, the conventional NK cells, or both cells, and then we can use these models then to compare responses at mucosal sites, and of course, for the NK cells, can look at NK cell biology in general in these mice. So I'll just show you some of the initial data we have with the IL-2 receptor gamma flox mice. They seem to be working pretty well. Here in the flox plus control, NK cells in the spleen. Here in the flox CRE mouse with the deletion of IL-2 receptor gamma in the NKp46 positive cells, an ablation of NK cells in the spleen. And similarly in the gut, we find a very strong decrease in the percentage of these NKp46 positive cells. And when we look for IL-22 induced expression in these NKp46 positive cells, we find that it's missing. So we think we have a model now where we can start to take apart these different subsets and try to identify roles for these different innate lymphoid subsets in the biology of infection. So just a final word on terms of stability. From Gerard's work, we know that the expression of RR gamma, at least in the subsets that express RR gamma, is a stable one. This compares with T cells, where we know that RR gamma expression can be lost as the cells exit the thymus. Many of these cells turn off RR gamma. But in the innate lymphoid cell population, these cells retain GFP expression. So all of the ILCs appear to stably express RR gamma, at least the subset that is an RRC positive subset. Now, with this NKp46 CRE model, we were able to start to look into the question related to plasticity and flexibility. And as we know, we're able to then tag these NK cells or tag these IL-22 producing cells by using this fate mapping approach. And when we do this experiment, by crossing again to this ROSA RFP and looking at the RFP positive cells in different tissues, we find that in most of the lymphoid tissues, including the thymus, the bone marrow, spleen, the lymph node, the NK cells that we find all stably express the CRE, meaning the GFP. So NCR expression is stable in those subsets, and we really don't have any loss of GFP expression in any of these NK cells that we find in these lymphoid tissues. But again, the gut is special, and when we look at the same type of analysis, only this time in the lamina propria, we see that we find a subset of cells that have lost GFP expression. So here, we're looking at, again, gating on these RFP positive cells, so cells that have expressed NKp46 or NCR1 in a previous life. Many of them continue to express NCR1, but a subset of them has lost expression of NKp46. And this can also be shown using antibody staining. It's maybe a little more clear. Here you have these IL-7 receptor positive NCR22 cells, the cells that we identify normally. Here we have the NK cells which lack the IL-7 receptor. These cells express NK11. And here we have the cells which have somehow lost expression of NKp46. So these are ex-NCR22 cells. And what's interesting is if you start to look at the functional capacity of these cells, we know that the NCR22 cells produce a lot of IL-22, but these cells appear to produce even more. So when you compare the levels of IL-22 production, these ex-NCR22 cells are actually producing much higher levels of IL-22, suggesting that in some way they've been modified in terms of their effector functions. Now, they also have some subtle differences in terms of phenotype. They remain RR gamma positive, but they lose expression of 2b4 and they become c-KIT positive. So there are some changes in the phenotype of these cells. But in terms of their functional output, they still remain IL-22 producing cells. They don't acquire IL-17. They don't acquire interferon gamma. So this kind of an approach, I think, is going to allow us to understand a little bit more about the interconversion, the flexibility of these different subsets. For the most part, it looks like the IL-22 ILC subset is very fixed in terms of its cytokine production. Its phenotype can obviously change, and now we have cells which can lose NKp46. We need to now go again and do the same kinds of analysis in these other ILC subsets to see if we really do have any level of plasticity and flexibility in this innate lymphoid cell family. And finally, of course, although much of this work is done in the steady state, we need to go back and be careful to look in cases where we have pathology. So inflammatory syndromes and potentially for especially these ILC2s that produce Th2-like cytokines, there may be some modifications of the biology of these cells under different pathologic conditions like allergy and asthma. And of course, the autoimmune disease models that we know in mucosal surfaces and in other tissues may be very important to really pin down the question of flexibility and interconversions between these different subsets. So I'll end there and just summarize. We think that the ILC subset is an emerging family. We think this represents a branch of the hematopoietic tree, which depends on ID2, requires ID2, again, to bring these lymphoid precursors into a new developmental pathway, which generates an innate lymphoid cell. We think these cells develop independently. They acquire their functions very early. And of course, now the big question is in terms of their effector functions and their redundant functions within different sites. And again, abundance of these cells at the mucosal site. The question is, of course, do they also exist in other tissues, and what role could they potentially be playing there under homeostasis or disease conditions? So this work was done by Naoko Sato Takayama in the laboratory. She's really been the driving force for the discovery of these NCR22 cells. I'd like to also mention Christian Vos-Henrich, who's generated these cream mice and has been quite interested in the questions related to NK cell development in the laboratory. A great collaboration with Shin and Gerard. We have collaborators as well providing different reagents like Jean-Christophe Renaud for the IL-22 work, Ofer Mandelbaum for the NCR-GFP mice, some nice collaborations with Nadine on the function of NK cells in infection, and work with Hergen that I didn't have time to mention. So thank you for your attention. I'm happy to take questions.